Scientists traced the plague's DNA back through 600 years of death, suffering, and historical transformation. What they found was not some ancient, extinct pathogen from a distant and inaccessible past. What they found was terrifying in its simplicity and its profound implications for modern disease understanding and pandemic preparedness. The bacteria that killed 75 million people across Europe, Asia, and North Africa in the 14th century is genetically almost identical to strains of Yersinia pestis that exist in the world today. The Black Death never died. It never went extinct. It evolved slowly. It adapted to changing conditions. It persisted in animal populations across continents. And it is still here, lurking in rodent colonies around the world, waiting for opportunities to infect human populations. This discovery fundamentally changed everything we thought we knew about one of history's most devastating pandemics and about the nature of pandemic disease itself. For centuries, historians and scientists working across multiple disciplines assumed that the plague of the Middle Ages must have been caused by a different, more virulent, more deadly strain of bacteria. Something unique to that historical time period. Something that had since gone extinct through natural selection or evolved into a less dangerous form through centuries of evolutionary pressure. The mortality rate seemed too astronomically high, the geographic spread too rapid and extensive, the clinical symptoms too severe and dramatic for it to be the same disease that occasionally appears in isolated cases in the modern world. There had to be something fundamentally different about medieval plague compared to modern plague. Some genetic quality or virulence factor that made it uniquely deadly compared to modern plague outbreaks that kill only dozens of people even in significant events. Then scientists extracted DNA from the teeth of plague victims buried in mass graves across Europe and beyond. From London's East Smithfield Cemetery. From Barcelona's medieval burial grounds. From Marseille's plague pits. From dozens of medieval cemeteries that had lain undisturbed for 6 centuries, sealed beneath layers of earth and urban development. They extracted precious bacterial DNA preserved in dental pulp, protected inside tooth enamel from centuries of decay and environmental contamination. They sequenced the entire genome of the Black Death bacterium gene by gene. They compared it systematically to modern plague strains collected from various parts of the world over the past century. And what they discovered through painstaking analysis shattered previous assumptions about pandemic disease and historical epidemiology and microbial evolution. The medieval plague was definitively Yersinia pestis. The same bacterial species that causes plague today in modern outbreaks in Madagascar, Mongolia, and the American Southwest. Not a different subspecies or related organism. Not a more virulent ancient variant that had since disappeared from the earth. The same bacteria at the species level. Genetically, when you compare the complete DNA sequences, the 14th century Black Death and modern plague strains are separated by less than 1% difference in their total DNA sequences. 1% genetic difference across the entire genome. 600 years of evolution in diverse environments. Millions of deaths across multiple continents and historical periods. Countless opportunities for mutation and selection. And the bacteria is essentially unchanged at the fundamental genetic level when you examine its core genome and virulence genes. The implications of this discovery are profound and deeply unsettling for our understanding of pandemic potential and disease evolution. The first major groundbreaking study came from Johannes Krauss and his interdisciplinary team at the University of Tübingen in Germany, working in close collaboration with researchers from McMaster University in Canada and University College London. They extracted DNA samples from the teeth of plague victims buried in the East Smithfield Plague Cemetery in London, a specialized mass burial site specifically created during the catastrophic years of 1348 and 1349 when normal burial practices completely broke down. The cemetery had been excavated during construction work in the 1980s, revealing hundreds of skeletons arranged in careful rows despite the overwhelming circumstances, powerful testimony to the overwhelming scale of death during those plague years and the attempts to maintain dignity even in catastrophe. The researchers focused specifically on teeth rather than bones because dental pulp provides an excellent and protected environment for DNA preservation over centuries and millennia. Bacteria that colonize the bloodstream during systemic infection can enter the pulp chamber through the tooth's blood supply and remain preserved there after death, sealed inside the hard enamel coating of the tooth that protects against environmental degradation and contamination. The extraction and sequencing process was extraordinarily difficult and required developing entirely new laboratory techniques and protocols. Ancient DNA is highly fragmented into tiny pieces, significantly degraded by time and chemical processes, contaminated with DNA from soil bacteria and fungi and from centuries of environmental exposure and from modern handling during excavation. The researchers had to develop new methodological approaches to extract minute quantities of bacterial DNA from centuries-old samples with minimal modern contamination. They had to distinguish genuine ancient DNA sequences from modern contamination introduced during excavation or laboratory processing using multiple authentication methods. They had to sequence millions of tiny fragments and computationally reconstruct the complete genome from thousands of tiny overlapping pieces like assembling a massive three-dimensional jigsaw puzzle with missing pieces. The technical challenges were immense and required years of methodological development and validation. But the results were absolutely worth the extraordinary effort and investment. They successfully reconstructed the complete genome of the Black Death bacterium from multiple individuals buried in the cemetery. Every gene in the bacterial chromosome. Every mutation that had accumulated. Every DNA sequence across the entire genome. They could now compare this medieval genome systematically to modern plague strains collected from various parts of the world over the past century and stored in laboratory collections. They could trace evolutionary changes over time with precision. They could identify which modern strains were most closely related to the medieval pandemic strain through phylogenetic analysis. They could build a detailed family tree of plague bacteria spanning 6 centuries of evolution and geographic distribution across multiple continents. The results were published in the prestigious journal Nature in October 2011 and caused immediate controversy and intense fascination in both scientific and historical communities worldwide. The medieval plague was definitively identified as Yersinia pestis beyond any reasonable doubt. Definitively proven through multiple lines of genetic evidence. Beyond any reasonable scientific doubt or alternative explanation. No question remaining about the identity of the pathogen. The same species that causes bubonic plague today in modern outbreaks across the world. The genome sequence was remarkably similar to modern strains analyzed from recent plague cases. The key virulence genes, the specific genetic sequences that make the bacteria deadly to humans, were present in the medieval strain and are present in modern strains in nearly identical form with only minor variations. The bacteria had the same basic biological toolkit for causing disease in the 14th century as it does in the 21st century. The genetic blueprint for causing plague was essentially unchanged. This raised an immediate and deeply troubling question that scientists and historians had to address. If the bacteria is essentially genetically identical at the DNA level, why did the medieval plague kill so many more people across such vast territories? Why did it spread so much faster across continents and through populations? Why was the mortality rate so much higher, killing perhaps half the population of Europe, compared to modern plague outbreaks, which typically kill fewer than a dozen people even in significant events? If the bacteria has not significantly changed at the genetic level over 6 centuries, something else must explain the dramatically different outcomes between medieval and modern plague epidemiology and mortality patterns. The answer lies not in the bacteria's genetic makeup but in human circumstances and medical capacity to respond. Medicine and medical technology. Public health infrastructure and disease surveillance systems. Scientific understanding of disease transmission and causation. Living conditions and urban sanitation. Population density and housing quality. Nutrition levels and baseline health status. All the environmental and social factors that determine whether exposure to plague bacteria results in death or survival. Medieval Europe had none of the advantages of modern medicine or public health infrastructure. No antibiotics or antimicrobial drugs. No understanding of bacterial infection or germ theory of disease. No public health measures based on scientific understanding of disease transmission. No rapid diagnostic testing to identify infections early. No isolation protocols for infectious cases based on transmission knowledge. No effective treatment options beyond bloodletting and prayer and herbal remedies that had no efficacy against bacterial infection. When someone in medieval Europe contracted plague through a flea bite or exposure to respiratory droplets, they almost certainly died within days to weeks. The mortality rate for completely untreated bubonic plague is approximately 60% even with basic supportive care like hydration and fever management. For pneumonic plague, the respiratory form of the disease that spreads person to person through coughing, the mortality rate approaches 100% without prompt antibiotic treatment started very early in infection. In the 14th century, there was no antibiotic treatment available. There was no effective treatment of any kind based on understanding of bacterial infection. There was no understanding that bacteria even existed as a biological concept. So plague killed at its maximum natural mortality rate determined by the bacteria's virulence, completely unchecked by any effective medical intervention that could interrupt the infection's progression. Today, plague is readily treatable with common antibiotics like streptomycin or gentamicin or modern fluoroquinolones like ciprofloxacin. If diagnosed early through clinical suspicion and laboratory testing and treated promptly with appropriate antibiotics at adequate doses, the mortality rate drops dramatically to less than 15% even for bubonic plague cases with good medical care. Pneumonic plague, if caught very early in infection within the first 24 to 48 hours and treated aggressively with intravenous antibiotics, can also be successfully treated, though the window for effective intervention is very narrow, only 1 to 2 days from initial symptoms to potential death without treatment. Modern medicine has not eliminated plague from the environment. The bacteria still exists in wild animal populations, particularly rodents and their fleas in parts of the American West, Sub-Saharan Africa, and Central Asia. But modern medicine has made plague survivable and containable in most cases with proper medical care and public health response. The genetic similarity between medieval and modern plague also revealed something else profoundly important about disease history and microbial evolution. The Black Death did not appear out of nowhere as a completely novel pathogen without precedent. It was not a new disease in the 14th century emerging suddenly from unknown sources. Yersinia pestis is ancient in evolutionary terms. Genetic analysis using molecular clock methods and evolutionary modeling suggests that the bacteria emerged as a distinct species from its less virulent ancestor thousands of years ago, possibly as early as the Bronze Age 3,000 to 5,000 years before present. There were probably earlier plague pandemics in ancient history that went completely unrecorded in surviving historical sources or were attributed to other causes by ancient writers who had no understanding of bacterial disease or epidemiology or transmission mechanisms. The plague of Justinian in the 6th century CE, which devastated the Byzantine Empire and killed an estimated 25 to 50 million people across the Mediterranean world and beyond, was almost certainly caused by Yersinia pestis based on contemporary historical descriptions of symptoms. Recent genetic studies have successfully extracted and sequenced plague DNA from 6th century burial sites in Bavaria and other locations across Europe, and the results definitively confirm that the Justinian plague was indeed caused by the same bacterial species as the medieval Black Death and modern plague, though from a different strain lineage that may have since gone extinct. The family tree of plague extends back at least 1,400 years of documented history through genetic evidence, probably much longer into prehistoric times before written records. This means that Yersinia pestis has been a significant human pathogen causing epidemic disease for a very long time, millennia spanning human civilizations, not just centuries of recorded history. It has been killing humans and shaping human history throughout recorded civilization. It caused multiple devastating pandemics across different historical periods and geographic regions from ancient times to the present. The medieval Black Death was not unique in being caused by this particular bacteria. It was just the most thoroughly documented historically through medieval chronicles and records, and the most devastating in terms of absolute death toll and percentage of total population killed. The bacteria has been with us throughout recorded history since the first cities and agricultural settlements, and probably for thousands of years before writing systems developed to record its catastrophic effects on human populations. Scientists trace the evolutionary history of Yersinia pestis systematically by comparing DNA sequences from different historical periods and different geographic locations worldwide where samples could be obtained. They built detailed phylogenetic trees showing how different strains are related to each other evolutionarily and when they diverged from common ancestors through accumulated mutations. They identified mutation rates in different lineages by comparing sequences and calculated evolutionary timelines using molecular clock methods that assume constant mutation rates. The complex picture that emerged was fascinating and scientifically intricate, revealing multiple plague pandemics over human history caused by different lineages of the same bacterial species. The Black Death pandemic appears to have originated in Central Asia, probably in the region that is now Mongolia or Western China or Kazakhstan based on genetic diversity analysis of modern strains. This is where the most ancestral and genetically diverse strains of the medieval plague bacteria are found in modern rodent populations that serve as natural reservoirs. From there, the bacteria spread westward along established trade routes during the 14th century, carried by infected rats and fleas traveling with merchant caravans along the Silk Road trade network. The Silk Road, that ancient highway of commerce connecting east and west through Central Asian oases and mountain passes, became an unexpected highway of death, carrying plague bacteria from Asian reservoir populations to the Middle East and eventually to Europe through multiple transmission chains and routes. The bacteria entered Europe through multiple independent routes simultaneously during the mid-14th century, not just through one single entry point or event. The famous Genoese merchant ships that fled from the siege of Caffa on the Black Sea coast and arrived in Sicily in October 1347 were one important vector of introduction that is well documented, but definitely not the only route. Overland trade routes through the Middle East and Anatolia brought plague to Constantinople and other Mediterranean ports through separate transmission chains involving different merchant networks. The disease spread from multiple entry points scattered across southern and eastern Europe simultaneously, creating a complex web of infection that quickly covered the entire European continent within 3 to 4 years from 1347 to 1351. The pandemic was not spreading outward from a single geographic point of origin in a simple wave. It was multiple overlapping waves from different introduction points converging and reinforcing into a continental catastrophe. Once successfully established in European rodent populations through these initial introductions, the bacteria adapted to local ecological conditions through natural selection and evolved into distinct regional lineages through random mutation. Different strains evolved in different geographic regions through random mutation accumulation and local evolutionary pressures. Scientists can now trace the geographic spread and evolution of plague across Europe by analyzing DNA from different burial sites and comparing the genetic sequences to identify transmission patterns and evolutionary relationships. They can see with remarkable precision how the bacteria moved from Sicily to mainland Italy, from Mediterranean ports to inland cities along river valleys and trade routes, from southern Europe to northern Europe and eventually to Scandinavia and even Iceland. Each region's plague victims carry bacterial DNA that tells a detailed story of how the disease spread geographically and evolved genetically through Europe over decades. The research also revealed something remarkable and unexpected about modern plague distribution globally. All modern plague strains found anywhere in the world today descend evolutionarily from the medieval Black Death pandemic of the 14th century. Every single strain of Yersinia pestis that exists today in any geographic location can trace its evolutionary ancestry back through genetic analysis to the 14th century pandemic that killed tens of millions. The Black Death was not just a singular catastrophic event in human history. It was an evolutionary bottleneck that fundamentally reshaped the global population structure of plague bacteria worldwide. Before the Black Death pandemic, there may have been considerably more genetic diversity in plague bacteria populations distributed across Eurasia. After the Black Death swept across continents, essentially all plague bacteria everywhere in the world are descendants of the strains that caused the medieval pandemic. Other lineages appear to have gone extinct. This has profoundly important implications for understanding how pandemics work from both evolutionary and ecological perspectives. The Black Death was so successful from the bacteria's evolutionary perspective that it essentially replaced all previous plague strain lineages globally through competitive exclusion. The descendants of the Black Death bacteria are the ones that survived and spread successfully and established themselves in rodent populations around the world from North America to Africa to Asia. The pandemic was not just a human catastrophe causing enormous suffering. It was also a bacterial evolutionary success story that allowed one particular lineage of Yersinia pestis to dominate globally and eliminate competing bacterial lineages through superior transmission or survival characteristics. The implications extend far beyond understanding historical plague. This research demonstrates that major pandemics can fundamentally reshape the genetic structure of pathogen populations globally, creating genetic bottlenecks where only certain lineages survive and spread. This has implications for understanding other pandemic diseases including influenza, where pandemic strains periodically emerge and replace existing seasonal strains, and for understanding emerging infectious diseases where founder effects and bottlenecks shape pathogen evolution during geographic spread. Scientists also discovered that the plague bacteria mutates relatively slowly compared to many other bacterial pathogens that cause human disease. The mutation rate of Yersinia pestis is demonstrably low when compared to rapidly evolving bacteria like those causing tuberculosis or antibiotic-resistant staph infections. This comparatively slow mutation rate explains why 600 years of evolution across diverse environments has not significantly changed the bacteria's genome structure or its basic pathogenic characteristics at the molecular level. Slow mutation accumulation means slow evolutionary change over time in the absence of strong selective pressures. The bacteria that killed millions of people in 1348 would be recognized as essentially identical at the DNA sequence level by the immune systems of people living today. Our human immune systems have not evolved specific protective immunity to plague despite 6 centuries of repeated exposure across multiple generations because the selection pressure was not sustained long enough, and the bacteria has not evolved into something significantly different despite the same length of evolutionary time because mutation rates are inherently low. This relatively slow mutation rate has practical and important implications for modern medicine and public health preparedness against plague outbreaks. It means that antibiotics that work effectively against plague strains today would almost certainly have worked against the medieval plague strains if those antibiotics had somehow existed in the 14th century. The bacteria has not evolved significant antibiotic resistance despite decades of antibiotic use in medicine because it mutates slowly and because human populations in modern times are no longer exposed to the sustained selective pressure of widespread antibiotic use specifically against plague since the disease is now rare. Plague is rare enough in modern populations that antibiotic resistance has not become a major clinical problem despite widespread antibiotic use against other bacterial infections. This is medically fortunate from a public health perspective, because antibiotic-resistant plague would be absolutely catastrophic given the bacteria's naturally high mortality rate and rapid progression without treatment. The comprehensive DNA studies also revealed fascinating and important details about how the bacteria causes disease at the molecular and cellular level through specific virulence mechanisms. Scientists identified specific genes in the plague genome that code for virulence factors, proteins and toxins that the bacteria uses to invade human cells, evade the immune system response, and cause the characteristic devastating symptoms of plague including buboes and septicemia. These virulence genes are present in both medieval and modern strains in virtually identical form with only minor sequence variations. The bacteria's basic biological strategy for causing disease through specific molecular mechanisms has not changed significantly over 6 centuries of evolution. The genetic toolkit for pathogenesis is conserved. One particularly important and well-studied virulence factor is the plasminogen activator gene called PLA, which allows plague bacteria to spread rapidly through tissue and break down blood clots that would otherwise physically contain and limit the infection. This powerful enzyme contributes significantly to the rapid bacterial spread throughout the body through the bloodstream and the severe hemorrhagic symptoms of advanced plague, including bleeding under the skin and from mucous membranes. Another key virulence factor system is the Yersinia outer proteins, or YOPs, which the bacteria injects directly into human immune cells using a molecular syringe called a type 3 secretion system to disable those cells and prevent effective immune response against the infection. These sophisticated molecular weapons evolved to defeat the human immune system are present in both medieval and modern plague bacteria in remarkably similar forms. The medieval Black Death bacteria had the same arsenal of virulence factors and pathogenic mechanisms that modern plague bacteria possess and deploy today against human hosts. Scientists also investigated systematically whether the medieval bacteria had any unique mutations or genetic elements that made it more inherently deadly or more transmissible than modern strains at the genetic level. They looked carefully for genes that might be present in medieval plague genomes but absent in modern strain genomes, which could explain higher virulence. They looked for genes present in modern strains but absent in medieval strains, which might indicate evolutionary attenuation. They looked for specific mutations in virulence genes that might affect the protein's function and thus virulence. They found very little significant difference in virulence-associated genes. The medieval bacteria was not measurably more virulent at the fundamental genetic level than modern bacteria when comparing virulence gene sequences. It was just encountering a completely immunologically naive and medically vulnerable human population with absolutely no medical defenses against bacterial infection or understanding of disease transmission. The genetic evidence definitively shows that pandemic severity depends critically on the interaction between pathogen characteristics and host population vulnerability rather than on pathogen genetics alone. The same bacterium with the same virulence genes can cause dramatically different outcomes depending on whether it encounters a medieval population with no antibiotics and no understanding of disease transmission, or a modern population with advanced antibiotics and sophisticated public health infrastructure. This is a crucial insight for pandemic preparedness. We cannot assume that historical differences in pandemic severity necessarily reflect differences in pathogen virulence at the genetic level. Context matters enormously. This research has profound practical implications for understanding and preparing for future pandemics that may threaten human populations. It demonstrates conclusively that pandemic potential and severity depend not just on inherent pathogen characteristics coded in DNA, but critically on host population vulnerability and medical capacity to respond effectively. A bacteria that causes only limited disease killing dozens in modern populations with ready access to antibiotics and sophisticated public health infrastructure can cause absolutely catastrophic pandemics killing millions in populations without those technological and organizational advantages. The bacteria itself may not change significantly over centuries at the genetic level, but the outcome of infection depends critically and decisively on the medical and social circumstances and infrastructure surrounding the infection event and disease spread. The genetic similarity between medieval and modern plague strains also means that we can use modern plague as a direct biological model for understanding the medieval pandemic in unprecedented detail beyond what historical sources alone could provide. We can study how modern plague bacteria cause disease at the cellular and molecular level in laboratory models and animal experiments, how they spread through populations in modern outbreaks, how they interact with the human immune system through immunological studies, and apply that hard-won scientific knowledge directly to understanding historical plague dynamics and outcomes. The biology is essentially identical at the genetic and molecular level. The fundamental difference between medieval and modern outcomes is in the medical and social context surrounding infection rather than in the pathogen itself. Scientists working in this field continue to refine and significantly expand this research program using new technologies as they become available. New burial sites are discovered and carefully excavated with modern archaeological methods that preserve ancient DNA. New DNA samples are meticulously collected from remains and sequenced using ever-improving genetic sequencing technologies that can handle degraded ancient DNA more effectively. Each new sample, each new sequence, adds incrementally to our collective understanding of how plague evolved and spread across continents and through time. The complete genetic and evolutionary history of one of humanity's greatest microbial killers is being painstakingly written and reconstructed, one precious ancient DNA sample at a time extracted from victims who died centuries ago. The research has also sparked renewed and intense interest in historical plague outbreaks beyond just the most famous Black Death of 1347 to 1353. Historians are systematically revisiting medieval and early modern records with new questions directly informed by genetic data and phylogenetic analyses. They are looking for evidence of how different strains of plague identified through genetics might have caused different outbreak patterns and mortality rates across regions. They are comparing historical descriptions of plague symptoms in contemporary chronicles and medical texts with modern medical knowledge of Yersinia pestis infection to validate and interpret historical accounts. The highly productive collaboration between geneticists, microbiologists, historians, and archaeologists has created a much richer and more nuanced and more scientifically detailed picture of the Black Death and subsequent plague pandemics than was previously possible with historical sources alone or genetic data alone. The interdisciplinary synthesis is greater than the sum of its parts. One particularly fascinating and important finding that has emerged from multi-site genetic analysis is that the plague appears to have entered Europe multiple independent times during the 14th century from different source populations, not just once in the famous 1347 introduction through Genoese merchant ships at Messina in Sicily. Genetic analysis comparing samples from different geographic regions and different burial contexts using phylogenetic methods suggests that there may have been several independent introductions of genetically distinct plague bacteria from Asian reservoir populations into Europe around the same general time period in the mid-14th century. Different introduction events from different source populations would have brought genetically distinguishable strains that can be identified through sequence comparison. This surprising finding would explain why the disease spread so extraordinarily rapidly across the European continent. It was not spreading outward from a single geographic point of origin in southern Italy but from multiple simultaneous entry points scattered across southern and eastern Europe including Italy, the Balkans, and possibly other regions. The pandemic was not a single wave emanating concentrically from one source location but multiple overlapping waves from different introduction events converging and reinforcing and creating a coordinated continental catastrophe that overwhelmed any possibility of containment. The DNA evidence recovered from burial sites also shows definitively that plague persisted endemically in Europe for centuries after the initial Black Death pandemic of 1347 to 1353 ended. There were repeated and devastating outbreaks, the so-called Second Plague Pandemic that continued into the 18th century with periodic and unpredictable resurgences every few years or decades. Genetic analysis of samples from these later outbreaks dating from the 15th, 16th, and 17th centuries clearly shows through phylogenetic comparison that they were caused by direct descendants of the original Black Death bacteria through continuous transmission. The disease had successfully established itself in European rodent populations, creating endemic foci, and kept resurging periodically whenever environmental and social conditions allowed efficient transmission from rodent reservoirs to human populations. Factors like climate fluctuations affecting rodent populations, urban crowding, wars and social disruption, and trade patterns all influenced when and where plague would resurge from its endemic animal reservoirs. The bacteria finally disappeared from Europe as an endemic disease in the 18th century through a combination of interacting factors that disrupted the transmission cycle, probably due to significant changes in urban living conditions and housing quality, improvements in housing construction and urban sanitation that dramatically reduced human contact with rats and their fleas through better building practices, and possibly evolutionary or ecological changes in the dominant rat species. Present in European cities. The black rat or roof rat, Rattus rattus, that primarily carried plague-infected fleas and lived in close association with humans was partially replaced ecologically by the brown rat or Norway rat, Rattus norvegicus, which lives in somewhat different habitats, often outdoors, and has different behavioral interactions with humans and different flea species. These ecological and social and architectural changes disrupted the ancient cycle of plague transmission between rodent reservoirs and human populations. The bacteria did not go extinct globally. It still existed and thrived in Asian and African rodent populations far from Europe, but it was effectively eliminated from European rodent reservoirs through changed ecological conditions. The last major European plague outbreak occurred in Marseille in 1720 to 1722, after which the disease disappeared from the continent for over 2 centuries. Today, plague persists as an endemic disease in wild rodent populations in geographically scattered areas across multiple continents, including North America, particularly the American Southwest, South America, particularly Peru and Bolivia, Sub-Saharan Africa, particularly Madagascar and Tanzania, and Asia, particularly Mongolia and Western China and Kazakhstan. It occasionally spills over from these wildlife reservoirs into human populations when people come into contact with infected rodents or fleas, causing small outbreaks that are quickly detected through disease surveillance systems and contained with modern medicine and public health surveillance and response measures. The bacteria causing these modern outbreaks is still genetically the same Yersinia pestis species that caused the medieval Black Death 6 centuries ago. It still has the same genetic toolkit and virulence factors for causing deadly systemic disease in humans through the same pathogenic mechanisms. But it encounters a profoundly different world with antibiotics that can kill bacteria, with sophisticated disease surveillance systems that can detect outbreaks early, with public health infrastructure specifically designed through decades of development to detect and rapidly contain infectious disease outbreaks before they spread widely. The bacteria fundamentally has not changed significantly at the genetic level. We humans have changed dramatically in our capacity to detect and respond to bacterial infections. This reality raises a sobering and uncomfortable question about pandemic preparedness for the future that policymakers must seriously consider. If social and medical conditions were to deteriorate significantly across large populations, through major regional or global war, severe and sustained economic collapse, catastrophic breakdown of public health systems due to prolonged crisis, could plague potentially resurge as a major killer of millions as it was in medieval times? The bacteria still exists widely in the environment in animal reservoirs. It still has demonstrated pandemic potential across centuries. Only our medical and social defenses built over decades keep it effectively in check and prevent it from causing mass mortality. Those defenses are not permanent or automatically guaranteed to persist. They require constant financial investment, institutional maintenance, trained personnel, and sustained political will across election cycles and generations. Any significant disruption to medical or public health infrastructure could potentially allow plague or other treatable diseases to cause casualties at levels not seen for centuries. The story of plague DNA research is fundamentally also a story about the remarkable power of modern scientific techniques and technologies to answer historical questions that seemed completely unanswerable and forever unknowable just a few decades ago. Before ancient DNA technology was developed starting in the 1980s and 1990s, we could only make educated guesses about what specific pathogens caused historical pandemics based solely on symptom descriptions in medieval chronicles and early modern medical texts. Now we can extract and sequence actual pathogen DNA directly from victims' remains that have been buried for centuries and identify the exact bacterial or viral species responsible with molecular precision. We can trace its evolution over time through phylogenetic analysis. We can understand its biology through comparative genomics. We can compare historical and modern strains directly at the DNA sequence level. This represents a genuine scientific revolution in historical understanding that has fundamentally transformed how we study disease history. The sophisticated laboratory techniques and analytical methods developed specifically for plague research have been successfully applied to many other historically important diseases as well. Scientists have extracted and sequenced DNA from tuberculosis causing Mycobacterium tuberculosis from ancient Egyptian mummies and medieval European remains, revealing the ancient origins and evolution of this major killer. They have recovered leprosy DNA from medieval skeletons, tracing how this disease spread through Europe. They have even recovered genetic material from historical smallpox outbreaks from carefully preserved specimens, providing insights into this now eradicated disease. Each successful extraction and sequence determination adds to our growing understanding of disease history and pathogen evolution across human civilization. The emerging field of paleomicrobiology, the systematic study of ancient microorganisms through genetic and other methods, has exploded in growth and importance in recent decades due to technical advances in DNA extraction and sequencing that make ancient DNA studies increasingly feasible and routine. The plague research also powerfully highlights the crucial importance of maintaining museum collections and carefully preserving archaeological sites for future research. The precious samples that allowed breakthrough plague research to occur came from museum collections of human skeletal remains that have been carefully curated, catalogued, and preserved for decades or even centuries in some cases. Without those preserved collections housed in museums and universities, maintained through institutional commitment and funding, the revolutionary research would have been completely impossible. This demonstrates clearly the scientific value of maintaining biological and archaeological collections, even when we cannot immediately see or predict how they might be useful for research. Future technologies not yet invented may allow us to extract types of information from carefully preserved specimens that we cannot even imagine or conceive today. Maintaining collections is an investment in future scientific discovery. The research program studying ancient plague DNA continues to evolve and expand in new directions as new questions emerge from initial findings and as new technologies become available. Scientists are now investigating whether there were consistent genetic differences between plague strains that caused primarily bubonic outbreaks with bubo formation versus those that caused primarily pneumonic outbreaks with person-to-person transmission through respiratory droplets. They are looking systematically at how the bacteria adapted evolutionarily to different geographic and climatic conditions across its range from tropical Madagascar to cold regions of Central Asia. They are studying the coevolution of plague and its various rodent hosts through comparative genomics of host and pathogen. Each new study conducted, each new sample analyzed, adds layers of understanding and nuance to this ancient microbial killer that has shaped human history for millennia. Some researchers are also investigating the fascinating question of whether human genetic changes occurred in response to plague exposure over centuries. Did populations that survived multiple devastating plague pandemics over generations evolve any degree of genetic resistance through natural selection? There is some tentative but intriguing evidence that certain immune system gene variants may have been selected for in European populations after the Black Death through differential survival. Genetic variants affecting innate immune response and inflammation may have influenced who survived plague exposure and who died, potentially leaving genetic signatures in modern populations. This is an active and controversial area of research that combines plague genetics, human population genetics, immunology, and evolutionary biology into an integrated interdisciplinary approach. The plague DNA research also has important broader implications for understanding other pandemics both past and future beyond just plague specifically. It shows convincingly that pandemic severity depends critically on the complex interaction between pathogen characteristics and host population vulnerability rather than on pathogen genetics alone. It demonstrates that the same pathogen with identical genes can cause dramatically different outcomes, from minor local outbreaks to civilizational catastrophes, in different social and medical contexts. It emphasizes that medical and public health infrastructure are absolutely crucial for pandemic response and that maintaining this infrastructure requires sustained commitment. These hard-won lessons apply broadly to infectious disease threats of all kinds, not just plague. The research has enabled development of significantly better diagnostic tests for plague that can save lives through early detection. By understanding the complete genome structure, scientists can identify specific genetic sequences that are unique to Yersinia pestis and not found in related but non-pathogenic bacteria. This allows development of rapid PCR-based molecular tests that can detect plague infection within hours rather than the days required for traditional bacterial culture. Early diagnosis is critical for successful treatment outcomes, especially for pneumonic plague, which progresses very rapidly from initial symptoms to death in just days without treatment. Better, faster diagnostics directly translate into saved lives through enabling earlier treatment initiation. Vaccine development efforts have also benefited substantially from the genetic research program. Understanding the molecular virulence factors and how plague bacteria interact with the human immune system helps identify potential vaccine targets, bacterial proteins that could elicit protective immune responses if presented to the immune system safely. Several experimental plague vaccines are currently in various stages of development and testing, though none are currently licensed for widespread general use. An effective plague vaccine that provides long-lasting protection would provide valuable insurance against both natural outbreaks from environmental reservoirs and potential bioterrorism scenarios using plague as a biological weapon. The genetic similarity between medieval and modern plague demonstrated by DNA studies means that any effective vaccine developed today would presumably have worked against and prevented the medieval Black Death if it had somehow existed then and been widely distributed. This is a sobering thought about the potential impact of medical technology on historical events. The story of tracing the plague's DNA through 6 centuries is ultimately a powerful story about human ingenuity, scientific progress, and the quest for knowledge about our past. We have developed the technological ability to extract detailed genetic information from centuries-old remains preserved in museum collections and archaeological sites, and use that precious information to understand history in unprecedented detail and with molecular precision. We can literally read the genetic code letter by letter of the bacteria that killed millions of our ancestors. We can trace its evolution through time by comparing sequences. We can understand why it was so deadly through molecular biology. And we can prepare scientifically for the possibility that it might threaten human populations again through improved diagnostics, treatments, and potential vaccines. This represents the power of science to illuminate the past and protect the future. Scientists traced the plague's DNA systematically through 6 centuries of evolution and found a bacteria remarkably unchanged by time at the genetic level. The medieval Black Death of the 14th century and modern plague cases today are caused by the same bacterial pathogen, genetically almost identical. The genetic code preserved in the teeth of medieval victims buried in mass graves tells us that pandemic potential is not just about the pathogen and its virulence genes, but critically about the vulnerability of the human population it encounters and their capacity to respond. The bacteria persists widely in rodent populations in the environment, waiting for opportunities to infect humans. Our best defense is the medical and public health infrastructure we have built over generations to recognize and treat bacterial infections. That infrastructure must be maintained and strengthened with sustained investment if we are to avoid repeating history's deadliest pandemic. The research continues with new samples, new sites, new questions. Every plague burial ground excavated, every tooth analyzed, every genome sequenced adds to our collective understanding. The map of plague's evolutionary history grows more detailed and complex. Scientists now study not just when and where plague spread, but how it interacted with human societies at the molecular level. They investigate climate impacts on plague outbreaks through climate proxies and historical records. They study how warfare and social disruption affected plague transmission patterns. They analyze how different regions and cities responded to plague and whether those responses affected outcomes. The interdisciplinary research program grows broader and deeper. Modern genetic techniques continue advancing at a rapid pace, enabling ever more sophisticated analyses of ancient DNA samples. Next-generation sequencing technologies can now extract and sequence DNA from even more degraded samples than was possible just a decade ago. Computational methods for reconstructing ancient genomes from fragmented DNA have improved dramatically. Statistical methods for detecting ancient DNA signatures and distinguishing them from modern contamination have become more robust and reliable. These technical advances mean that samples once considered too degraded or contaminated to analyze can now yield usable genetic data. Archaeological sites once passed over as unlikely to preserve ancient DNA are being re-evaluated with new technologies. The field is expanding rapidly. The geographic scope of plague DNA research has also expanded significantly beyond the initial focus on European medieval burial sites. Scientists have extracted and analyzed plague DNA from victims in China, India, and other parts of Asia, tracing how the disease spread through Asian populations. They have analyzed samples from African plague outbreaks of the early 20th century. They are beginning to extract plague DNA from colonial American sites, investigating when and how plague reached the New World. Each new geographic region analyzed adds pieces to the global puzzle of plague evolution and spread. The picture becomes more complete with each new sample. Climate science has intersected productively with plague DNA research to understand how environmental factors influence plague outbreaks historically. By combining genetic data showing when plague was present with climate proxies showing temperature and precipitation patterns, scientists can investigate whether climate variations triggered plague outbreaks. There is evidence that climate conditions affecting rodent populations, particularly wet periods that increased rodent food supplies followed by dry periods that drove rodents into closer contact with humans, may have influenced plague outbreak timing. Understanding these environmental triggers could help predict future outbreak risks as climate continues changing. If warming temperatures expand the geographic range of plague-carrying rodents and their fleas northward, previously plague-free regions might become vulnerable. Monitoring environmental conditions and rodent populations becomes increasingly important for early warning. The ecology of plague has become clearer through genetic studies combined with field research on rodent populations. Plague naturally circulates in wild rodent populations in certain geographic regions called plague foci. Within these foci, the bacteria cycles between rodents and fleas in a sylvatic or wild cycle. Rodent populations expand and contract based on environmental conditions and disease prevalence. When rodent populations are high and plague is circulating intensely, spillover into human populations becomes more likely. When rodent populations crash due to plague mortality, the bacteria persists at low levels in resistant rodent species and their fleas, waiting for population recovery. This complex ecological cycle determines when and where human plague cases occur. Understanding these dynamics through ecological and genetic studies helps predict outbreak risks. Different rodent species have different roles in plague ecology. Some species like prairie dogs in North America are highly susceptible and experience massive die-offs during plague outbreaks, serving as sentinel species whose deaths indicate plague presence. Other species like deer mice are more resistant and can maintain plague bacteria in their populations without dying in large numbers, serving as long-term reservoir hosts. Understanding which species maintain plague and which species bring plague into contact with humans is crucial for disease ecology and prevention. Genetic studies of plague bacteria from different rodent hosts show how the bacteria adapts to different host species and which rodent species are most important for human transmission risk. Flea species also vary in their efficiency at transmitting plague from rodents to humans. The oriental rat flea is particularly efficient at transmission because it becomes physiologically blocked after consuming plague bacteria, forcing it to feed repeatedly in attempts to get blood, thereby inoculating multiple hosts with bacteria. Other flea species may carry plague bacteria but transmit less efficiently. Understanding flea ecology and behavior is crucial for understanding plague transmission dynamics and for developing control measures. Flea control around human habitations in plague-endemic areas is an important prevention strategy that requires understanding which flea species are present and how they interact with rodent hosts and humans. Human behavioral factors also play crucial roles in plague transmission that genetic studies alone cannot reveal. How humans interact with their environment, how they construct housing, how they store food, how they manage waste, all these factors determine how much contact occurs between humans and plague-carrying rodents and fleas. In modern Mongolia, for example, plague cases are associated with hunting marmots for fur and meat, bringing hunters into direct contact with infected animals. In other regions, poor housing quality that allows rodents to nest inside human dwellings increases exposure risk. In parts of Africa, storing grain in traditional granaries that attract rodents creates transmission opportunities. Understanding and modifying these behavioral and environmental risk factors is as important as understanding the bacteria's genetics for plague prevention. The bioterrorism potential of plague has been a concern since the biological weapons programs of the Cold War era. Both the United States and Soviet Union developed plague as a potential biological weapon before such programs were officially ended by international treaty. The Soviet program in particular developed antibiotic-resistant strains of plague and methods for weaponizing the bacteria for aerosol dissemination. While these specific weapon strains are believed to have been destroyed after the Soviet Union collapsed, the knowledge of how to create such weapons persists. The genetic similarity between natural plague strains and historical plague means that any naturally occurring plague bacteria could theoretically be modified and weaponized by a sophisticated program. However, the actual threat level from plague bioterrorism is debatable among security experts. Creating an effective biological weapon is technically challenging, requiring sophisticated laboratory facilities and trained microbiologists. Disseminating plague bacteria as an aerosol that would effectively infect large numbers of people is difficult. Modern disease surveillance systems would likely detect unusual plague cases quickly, enabling public health response including prophylactic antibiotic treatment of exposed individuals. The availability of effective antibiotic treatment means plague is not an ideal bioweapon compared to diseases like smallpox for which no effective treatment exists. Nevertheless, plague appears on the CDC list of Category A bioterrorism agents considered highest threat. Maintaining stockpiles of antibiotics and developing rapid diagnostic tests are considered important biodefense measures. Natural plague outbreaks continue occurring globally despite medical advances. Madagascar experiences hundreds of cases annually, with occasional deaths especially in remote areas where medical care is not immediately accessible. The United States sees a handful of cases each year in the Southwest, usually associated with flea bites from infected prairie dogs or rock squirrels. These cases remind us that plague has not disappeared and continues circulating in natural animal reservoirs waiting for opportunities to infect humans. Maintaining surveillance systems to detect cases rapidly and ensuring healthcare workers can recognize and treat plague are ongoing public health challenges even in wealthy countries with advanced medical systems. The global distribution of plague reflects historical trade patterns and rodent movements over centuries. Plague reached North America in the early 1900s through rats on ships from Asia arriving at California ports. The bacteria jumped from ship rats to native ground squirrels and other rodent species, establishing plague foci in the western United States that persist to this day. Plague reached South America through similar shipping routes. Madagascar likely acquired plague from ships arriving from India in the late 1800s. Each introduction established new plague foci in local rodent populations. Understanding these historical introductions through genetic analysis of modern plague strains from different regions reveals how human activities including trade and transportation have spread disease globally. These patterns offer lessons about how diseases can be transported across oceans and continents through global commerce networks. Climate change is expected to affect plague distribution and transmission dynamics in complex ways. Warming temperatures in some regions may allow plague-carrying rodent species and their fleas to expand their range northward into areas currently too cold for year-round plague transmission. Changes in precipitation patterns may affect rodent population dynamics, potentially increasing rodent populations in some areas and decreasing them in others. These ecological changes could alter the risk of human plague exposure in various regions. Some models predict increased plague risk in parts of Africa and Asia as climate warming allows plague-carrying fleas to survive in areas currently marginal for plague. Other models suggest plague risk might decrease in areas that become too dry to support rodent populations. The uncertainty about climate impacts on plague highlights the need for continued environmental and disease surveillance. One health approaches that consider the connections between human health, animal health, and environmental health are increasingly recognized as important for plague control and prevention. Plague is fundamentally a zoonotic disease that originates in animal populations, so controlling plague in human populations requires understanding and managing plague in rodent reservoirs. This requires collaboration between human medicine, veterinary medicine, wildlife management, and environmental science. Monitoring rodent populations for plague presence provides early warning of human risk. Controlling rodent populations around human settlements reduces transmission opportunities. Protecting pets from fleas that might carry plague from wild rodents reduces another transmission pathway. These integrated approaches are more effective than focusing solely on treating human cases after they occur. International cooperation on plague surveillance and control has improved significantly since the establishment of the World Health Organization and other international health agencies. Countries report plague cases to WHO, which tracks global plague occurrence and can assist countries with outbreaks through technical support and resources. International standards for plague diagnosis and treatment help ensure consistent medical care globally. Research collaborations share genetic data and epidemiological information internationally, enabling better understanding of plague ecology and evolution worldwide. This international cooperation is crucial for diseases like plague that do not respect borders and can spread through international travel and trade if not controlled. The ethical dimensions of ancient DNA research have received increasing attention as the field has grown. Extracting DNA from human remains raises questions about consent and respect for the dead. Many traditional communities have concerns about scientific research on their ancestors' remains. Professional ethics codes now emphasize the importance of consulting with descendant communities and respecting their wishes regarding research on ancestral remains. Repatriation of remains to communities requesting them is increasingly common. These ethical considerations must be balanced against the scientific value of the research. In some cases, communities have supported plague DNA research specifically because understanding historical disease impacts on their populations has contemporary value for health and heritage understanding. The communication of plague DNA research findings to public audiences has been challenging but important. The discovery that plague still exists and is genetically similar to the medieval Black Death can be frightening to non-scientific audiences who may not understand that modern medical capacity makes plague far less threatening today than it was historically. Science communicators must carefully explain both the genuine continuing risk from plague and the reasons why catastrophic medieval-scale pandemics are unlikely to recur given current medical capabilities. Balancing honest assessment of risk with avoiding unnecessary panic requires nuanced communication that respects public intelligence while explaining complex science accessible. The economic impacts of historical plague pandemics were staggering and transformed European societies fundamentally. The massive population losses in the 14th century disrupted labor markets completely, giving surviving workers unprecedented bargaining power. Feudal social structures that had lasted centuries crumbled as labor scarcity made traditional obligations impossible to enforce. Wages increased dramatically as lords competed for scarce workers. Land values fell as agricultural production declined. Long-distance trade patterns shifted as some commercial centers were devastated while others emerged stronger. Understanding these economic transformations through historical records combined with genetic evidence of when and where plague struck provides integrated pictures of how disease shaped societies through demographic and economic disruption. Cultural impacts of plague were equally profound and long-lasting. The massive mortality and apparent randomness of who died and who survived shook medieval religious faith and authority. Why did God allow such suffering? Why did prayers and religious devotion not protect the faithful? These theological questions haunted survivors and contributed to religious movements that challenged church authority. Art and literature became preoccupied with death and mortality. The danse macabre or dance of death became a recurring theme showing death claiming people of all social classes. Tomb sculptures increasingly featured transi tombs showing corpses in various states of decay. This memento mori tradition reminding viewers of mortality's inevitability reflected the psychological trauma of experiencing such massive death. These cultural transformations persisted for generations after the plague receded, showing how pandemic disease leaves psychological and cultural scars long after biological recovery occurs. Political consequences of plague outbreaks shaped European state development. The demographic catastrophe weakened some kingdoms while strengthening others based on how severely they were affected and how effectively their governments responded. Wars were interrupted or postponed as armies were decimated by plague. Political alliances shifted as some ruling families died out completely while others survived. The labor shortages created by plague ultimately contributed to peasant revolts in several countries as workers demanded better conditions and resisted attempts to reimpose traditional obligations. These social upheavals forced political changes, including increased centralization of power in some kingdoms. Understanding plague's role in these political transformations requires combining genetic evidence of disease timing and spread with historical analysis of political developments. The Renaissance emergence in Italy following the Black Death may have been partly enabled by the economic and social disruptions plague caused. The massive wealth transfers as plague victims died without heirs created newly wealthy families seeking to display their status through artistic patronage. The breakdown of traditional social hierarchies created social mobility, allowing talented individuals to rise regardless of birth. The questioning of traditional authorities, including religious authority that plague provoked, may have created intellectual climate more open to classical learning and humanist philosophy. While plague alone did not cause the Renaissance, it created conditions that made cultural transformation more possible. Understanding these complex historical connections requires interdisciplinary research combining history, genetics, economics, and cultural studies. The Protestant Reformation a century after the Black Death may also have been influenced by plague's cultural legacy. The questioning of church authority that plague provoked, the weakening of church economic power through property losses, the spread of vernacular literacy that labor scarcity encouraged, all these plague-related changes created conditions more favorable to religious reform. Martin Luther's emphasis on individual faith rather than institutional mediation resonated in populations that had seen religious institutions fail to protect them from plague. The printing press that spread Reformation ideas so rapidly was partly made economically viable by the literacy increases that occurred as education became more accessible after plague reduced population pressure on resources. These indirect effects of plague on later historical developments show how pandemic disease can have consequences that ripple through centuries. Colonial expansion from Europe beginning in the 15th century was partly enabled by the demographic recovery and economic transformation that followed plague. The population pressures that contributed to colonial expansion were partly reactions to the demographic collapse and recovery cycle that plague caused. The technologies and institutions that enabled colonial ventures were partly developed during the economic transformations following plague. The disease itself traveled with colonial expansion as European diseases, including later plague outbreaks, decimated indigenous American populations without immunity. Understanding these connections between medieval plague and later colonial impacts requires long-term historical perspective combining demographic, economic, technological, and epidemiological analyses. The scientific revolution that transformed European thought beginning in the 17th century may also have connections to plague's legacy, though these are debated among historians. The failures of traditional medical theory to explain or treat plague contributed to questioning of ancient authorities, including Galen and Aristotle. The empirical observation that quarantine and isolation measures seemed to help control plague even without theoretical understanding encouraged practical experimentation over theoretical speculation. The development of scientific methods emphasizing observation and experiment over authority may have been partly stimulated by plague's challenge to traditional medical knowledge. While plague alone did not cause the scientific revolution, it created intellectual conditions questioning traditional authorities that made new approaches more acceptable. Modern epidemiology as a scientific discipline has deep roots in responses to historical plague outbreaks. The observation that plague seemed to spread from person to person or through contact with infected materials contributed to early contagion theories even before germ theory. The practice of quarantine developed specifically for plague control. The systematic collection of mortality statistics during plague outbreaks laid groundwork for later epidemiological methods. John Grunt's analysis of London plague death records in the 1660s is considered foundational to statistical epidemiology. Understanding how historical plague control efforts contributed to modern epidemiology shows how disease challenges drive scientific development. Public health as an organized government function also has roots in plague responses. Medieval and early modern governments developed quarantine regulations, health boards to oversee plague control measures, and systems for isolating infected individuals. These institutional innovations established precedents for later public health agencies. The principle that governments have responsibility for protecting population health was partly established through plague control efforts. Modern public health infrastructure, including disease surveillance, quarantine authority, and isolation facilities, can trace its origins to institutional responses to historical plague. Understanding this institutional heritage provides perspective on current public health debates about government authority and individual rights during disease outbreaks. The concept of contagion itself was partly developed through observations of plague transmission patterns even before microorganisms were discovered. Physicians and public officials observed that plague seemed to spread from sick to healthy individuals, that avoiding contact with sick people reduced infection risk, that quarantining travelers from plague-affected areas helped protect communities. These practical observations contributed to contagion theories that competed with miasma theories attributing disease to bad air. The eventual acceptance of germ theory in the 19th century validated what plague observations had suggested centuries earlier, that diseases could be transmitted through material particles. Understanding this history of disease theory development shows how practical experience with pandemic disease gradually shaped scientific understanding. The discovery of Yersinia pestis as the cause of plague in 1894 by Alexander Yersin during the Hong Kong plague outbreak represented a major advance applying new bacteriological methods to understanding disease. Within a decade of this discovery, it was determined that rat fleas transmitted plague from rats to humans, providing the complete epidemiological picture. These bacteriological discoveries vindicated the empirical observations from centuries of plague experience that rats were somehow associated with plague and that avoiding infected materials reduced risk. Understanding how long it took to move from practical observations to scientific understanding provides perspective on how scientific knowledge develops gradually through accumulating evidence and technical advances. The development of antibiotics effective against plague in the mid-20th century transformed plague from inevitably fatal to usually treatable. The discovery that streptomycin could treat plague in 1947 provided the first effective treatment in thousands of years of plague history. This medical advance fundamentally changed plague's threat level, though access to antibiotics remains unequal globally. Understanding how recently effective plague treatment became available provides perspective on how modern medical capacity represents historically unprecedented ability to treat bacterial infections. This same perspective applies to many bacterial diseases that were major killers throughout history but became treatable within living memory through antibiotic development. The ongoing challenge of antibiotic resistance makes the lack of resistance in plague bacteria fortunate but not guaranteed to persist. While current plague strains remain antibiotic susceptible, the possibility of resistance emerging either naturally or through intentional modification remains concerning. Maintaining antibiotic effectiveness through responsible use policies, having alternative treatments available, and developing new antibiotics effective against potential resistant strains are important preparedness measures. The plague DNA research showing genetic stability over centuries suggests natural resistance emergence may be slow, but intentional modification for biological weapons purposes remains theoretically possible and concerning to security planners. Vaccine development for plague has been pursued since the late 19th century with varying success. Early killed whole cell vaccines provided some protection but had significant side effects and required repeated boosters. More recent vaccine development efforts have focused on subunit vaccines targeting specific plague antigens that might provide better protection with fewer side effects. Several experimental vaccines have shown promise in animal models and limited human trials, but no plague vaccine is currently licensed for general use in most countries. Some military forces maintain plague vaccines for personnel who might be at special risk. Further vaccine development is considered important for both natural outbreak preparedness and biodefense readiness, though the relatively small market for plague vaccines limits commercial development incentives. The integration of ancient DNA data with historical records and archaeological evidence has created much richer understandings of specific historical plague outbreaks than any single data source could provide alone. For example, the combination of genetic data showing when plague arrived in different locations with historical records of outbreak timing and archaeological evidence of mortality from burial patterns provides detailed pictures of how specific outbreaks unfolded. This integration reveals aspects invisible to any single approach. Genetics alone cannot tell us how societies responded. History alone cannot definitively identify disease causation. Archaeology alone cannot provide precise dates. The combination creates powerful integrated understanding exceeding what any discipline could achieve independently. Future directions for plague DNA research include even more geographically widespread sampling to understand global plague evolution more completely. Regions of Asia where plague first emerged remain undersampled compared to Europe. Africa's plague history is incompletely understood genetically despite ongoing plague transmission there. Sampling from historical plague outbreaks in different time periods would provide finer temporal resolution of plague evolution. Integrating plague genetics with ancient human DNA from the same individuals could reveal genetic factors influencing plague susceptibility or resistance. These future research directions will continue expanding understanding of plague's history and biology. The technical challenges of ancient DNA research continue driving methodological innovations applicable beyond plague studies. New extraction techniques maximize DNA yield from difficult samples. New sequencing approaches handle severely degraded DNA more effectively. New computational methods distinguish ancient DNA from contamination more reliably. New authentication methods verify ancient DNA authenticity more rigorously. These methodological advances developed for plague research benefit all ancient DNA research, including studies of human evolution, animal domestication, and other pathogen evolution. The investment in plague research thus produces broader scientific returns through methodological innovation. The interdisciplinary nature of plague DNA research demonstrates how major scientific questions increasingly require collaboration across traditional disciplinary boundaries. Understanding plague fully requires combining molecular biology, microbiology, genetics, evolutionary biology, ecology, history, archaeology, anthropology, climate science, and more. No single discipline has all necessary tools and perspectives. The most productive research emerges from genuine interdisciplinary collaboration where different specialists contribute unique expertise to shared questions. This model of collaborative interdisciplinary research applies increasingly to complex questions in many scientific fields beyond plague studies. The public engagement aspects of plague research are increasingly recognized as important for both science education and policy support. Ancient DNA research on historical disease is inherently interesting to general audiences who want to understand how their ancestors experienced disease and how modern medicine has changed human disease experience. Museums increasingly feature plague DNA research in exhibitions connecting historical artifacts with modern scientific understanding. Educational programs use plague DNA research to teach both historical and scientific content. This public engagement builds support for continued research investment and demonstrates science's value for understanding human experience beyond just practical applications. The preservation of plague DNA in archaeological remains for 6 centuries demonstrates that genetic information can persist much longer than previously thought possible. This realization has opened possibilities for ancient DNA research on much older specimens, including Neanderthal remains tens of thousands of years old, and ancient plant and animal remains. The methodological lessons from plague DNA research have enabled this expansion to deeper time depths. Understanding that DNA can preserve for millennia under right conditions has transformed paleontology, archaeology, and evolutionary biology by making genetic information accessible from extinct species and ancient populations. Scientists traced the plague's DNA through 6 centuries and discovered a bacteria remarkably unchanged while human societies transformed utterly. The medieval Black Death and modern plague are caused by the same pathogen genetically. The genetic code in medieval victims' teeth tells us that pandemic potential depends on vulnerable populations encountering pathogens, not just on pathogen genetics. The bacteria persists in rodent reservoirs worldwide. Our defense is the medical infrastructure we built. That infrastructure requires maintenance. The plague is still here. We are prepared. For now.